So next important is what are the applications of genetic engineering. So production of proteins in abundance, insulin, growth hormone, erythropoietin, interferons, blood clotting factors, vaccines, superoxide dismutase, all these can be produced in abundance. So insulin is very important in treatment of diabetes. So human insulin that is produced by recombinant DNA technology. Second most important is molecular analysis of diseases, genetic diseases, sickle cell anemia, thalassemias, familial hypercholesterolemia. So they can be analyzed, molecular analysis can be done. Then laboratory diagnostic applications for rapid diagnosis of diseases such as AIDS, we can do recombinant DNA technology. Then prenatal diagnosis of diseases, DNA collected from amniotic fluid can be used for predicting risk of developing genetic diseases by using DNA probe. So for example, sickle cell anemia also can be diagnosed, prenatal diagnosis. Then applications in forensic medicine, so to identify criminals, to settle disputes of parenthood of children, DNA fingerprinting, applying southernmost technique we can use. Then most important in gene therapy, so 1990 patient with adenosine deaminase deficiency was successfully treated with gene therapy. Gene therapy can cure several diseases such as sickle cell anemia, thalassemias, enzyme deficiencies. So we can use this technology in gene therapy. So there are a lot of industrial applications. Enzymes synthesized by recombinant DNA technology are used to produce sugar, cheese, detergents. Certain protein products are used as food additives to increase nutritive value and flavor. Then eighth application is agricultural applications. So genetically engineering plants, genetically engineered plants have been developed to resist drought and disease besides the higher crop yield. So BT cotton. Then transgenesis, so refers to transfer of genes into the fertilized ovum, which will be found in somatic as well as germ cells and passed on to the successive generation. Some success has been achieved in this regard in mice experiments. Last most important is in evolution. Recombinant DNA technique is great use in bridging several missing links in the evolution. So this is done by amplifying DNA from archaeological sample of extinct animals. So these are various uses of recombinant DNA technology. So we'll again revise in brief. So production of proteins in abundance, that is insulin, growth hormone, erythropoietin, interferons. Then molecular analysis of diseases such as sickle cell anemia, thalassemia. Then prenatal diagnosis, for example, sickle cell anemia. Then laboratory diagnosis of various diseases such as AIDS. Then application in forensic medicine, so identifying criminals, disputed paternity, this can be used. Then gene therapy for various diseases such as sickle cell anemia, thalassemia, enzyme deficiencies. Recombinant DNA technology can be used. Then there are various industrial applications. So recombinant DNA technology is used to produce sugar, cheese, detergents. Then agricultural applications, genetically engineered plants, they have been developed to resist drought. And transgenesis, so the transgenesis is also there. And last most important is in evolution. Recombinant DNA technology is very useful in bridging several missing links in evolution. So this is done by amplifying DNA from archaeological sample of extinct animals. So students, in today's lecture we have discussed in detail regarding what are the applications of recombinant DNA technology and what is the procedure of recombinant DNA technology. So LAQ, that is long question, is asked. Describe recombinant DNA technology in detail. So you have to write all 4 steps. You have to draw diagram, and there can be a short note of 5 marks applications of recombinant DNA technology. So 10 applications you have to write down. In long question also they can add a note on briefly applications. So you can write down